Okay. After we have the rating, then we go for the post announcer while going all faculty. Yeah, yeah, yeah. Thank you, Kailash. Sound is miss. Hmm. Like, I'll just. Okay, no, I'll just. I request my colleagues in DCS to sort of go to their own WhatsApp group and please call the students right now. कहाँ है तुम्हारे दोस्त, कहाँ है फर्स्ट ईयर के? और बस उतने मैसेज करो. हाँ हाँ. Going to start. I hope everybody told their students. Okay, so it's actually a pleasure to host Professor Subi George for this NSF colloquium. And Subi already has given one colloquium previously, maybe 6 years back, close to 6 years back. So it's a We welcome you again, Subi. So before we start the colloquium, I would like to state, maybe Subi has this idea already, that the Wednesday colloquium was started by Professor Homi Bhabha, the founding director. And the idea was he would visit on Wednesday and meet all the faculty members that day. And usually there would be a talk organized so that everybody gets together around him and listen to one of the experts from any part of the world or India visiting us that day. So, so all the events were organized so that he's there and can have discussion. So this has been a very, very prominent effort, academic effort in the institute. Everybody sort of is aligned to it. Nowadays, of course, it's hard to get people, but because of multiple seminar series. But the idea being that since it's a colloquium, physicists, chemists, and biologists all sit together and listen. And it has a broader appeal and enables students to crosstalk, even the PIs to crosstalk, as well as with the speaker, the expert. So for all of you, I would like to welcome to Professor Subi George colloquium. It's part of the TIFR Annual Chemistry Conference, the TACC, which is organized this year by Dr. Kalaivanan Nagarajan, who is a young faculty himself in this institute. And thank you, Kalai, for putting this efforts. And within the context, I would like to now introduce Subi. And it's a kind of a heavy introduction, although Subi doesn't seem, he has a very unassuming personality, but Subi has been long known for his supramolecular polymer work, supramolecular polymerization work, as well as organic electronics work. And some of this new ideas that are coming up from his lab is ingrained in the training that he received. First, of course, he did his bachelor's and master's in chemistry from Mahatma Gandhi University in Kerala, and then moved within the state to Trivandrum at the NIIST, where he worked with Professor Ajay Ghosh. And this is where Subi's ideas of, crystallization, non-covalent interaction, supramolecular structure all arose, albeit with smaller structures. But at that level of the complexity, he was already getting a handful ideas and trying to master very basic non-covalent interactions. He then jumped the ship from India and moved to Eindhoven University of Technology in Netherlands, and worked with a leader who was coming up with new ideas. Bert Meyer was coming up with new ideas of supramolecular polymer formation, polymerization. We all know that covalent polymers are very well known, but supramolecular polymerization was that time a new topic. And Subi gladly picked that up based on the expertise he had already developed in NIIST, and then came back to India and joined JNCASR in Bangalore. And from JNCASR, almost now, he has been in 15 years now, I think, right there in that institute, has been leading a fantastic world recognized research program, and also shaping that institute in itself by his own contributions, academic and non-academic contributions. Part of the fun with Subi is that he also has a very deep appreciation for other kinds of chemistry, and is always a fantastic guy to talk to. Because of all these accumulated success, and it's wonderful that we have one guy who represents us in many places. He has been awarded the Shanti Swarup Bhatnagar Prize in the COVID year, that was 2020, and very recently he is currently the associate editor in chemical sciences, a leading journal space in the chemistry community. There are many awards that Subi has won. I just mentioned these two, but suffice to say, he has been leading our front face from the country, leading the way on supramolecular polymerization ideas, the new ideas that are coming up in the community. So I welcome you again for this colloquium.
Thanks, JD, for a very, very kind introduction. It's really overwhelming. So good evening to one and all. And at the outset, let me thank, it's a great honor for me to be here and to be with all of you and share some of our recent results. So at the outset, let me thank Kalai, JD and Sudipta for a very kind invitation, and all the chemistry colleagues and all others from other departments. So it's so what I'll be doing is to share some of our recent results, which we have been doing in the last couple of years, basically, on recent results. But considering the diversity in the audience, what I will be doing is first probably 15 minutes, I'll be introducing the topic. So we are working on a kind of materials called supramolecular polymers. So this is probably during the last 2 decades, how, as JD mentioned, have emerged as one of the most promising material, soft material, if we want to think about a kind of adaptive and recyclable materials. Probably this is one of the well-known class of systems. So we have been lucky to grow along with the field. So what we have been trying to do is to address to increase the complexity and also to advance this field forward. So they're coming up with new concepts. So this is something which I'll be sharing with all of you in the next few minutes, next probably 15 minutes or so. So just to introduce to all of you what is the kind of material I'll be talking about, I'll be showing an animation. Yeah.
Escape.
Oh, sorry. I can use it. It's okay.
No, just a second. Got it. What is this?
It was working, but that.
Wait. It's here. Now. Now do it.
No, it's okay, JD. I'll manage. JD, it's okay. No, it's stuck. It's completely stuck. Machine over. Okay.
You have another discussion on this.
Let's check it once. Yeah, thank you. Sorry for the interruption. So just to give an introduction of the kind of materials we'll be working on, this is a short animation with a well-known class of molecules which many people have worked on. It's a simple benzene system with 3 amide groups that can form very weak interactions like hydrogen bonding, which can form one-dimensional structures. So as you can see that if you look into these structures, very similar to the macromolecules, which is polymeric structures. It consists of repeating units, many monomeric units, but the only difference is that unlike the classical covalent polymers, the only difference is here the monomers are attached through very weak interactions. So as a result, there will be always a kind of exchange dynamics involved in the system. So the monomers can go out of the backbone and come back. So this gives this kind of materials kind of reversibility, adaptivity, and self-repairing nature and so on. So this is the kind of materials I'll be talking about it. So over the last 2 decades, what has happened in this field is there is a lot of deeper mechanistic insight has happened how these polymer structures are grown, supramolecular polymer structures are grown. And especially during the last 5 years, a lot of attention has been paid to improve the precision synthesis of this system so that we get a kind of control on its structure. In terms of primary structure, especially, what I mean is in terms of is controlling its molecular weight, can we make them disperse, highly monodisperse system, just like classical, the protein and the biological structures and so on. And very recently, along with our group, many other groups are trying to push the limits of this self-assembly to go to more than one kind of multi-component systems and try to get a kind of sequence control. And we are very recently, today's talk, we'll be talking about how to control the next higher hierarchical level of this molecular self-assembly. Can you go to higher order structures and so on? So if you look into the list structure, again, coming back to this structure, as I mentioned, it's look like classical covalent polymers, macromolecules, and also very similar to the protein assemblies. So, okay, in dynamics, if you look into the dynamics, it's very similar to the biological structures, but in terms of structure, it's very similar to the classical polymers. So what we have been trying to do during the last 1 decade is to try to improve the control on this structure and also is temporal control. But I'll be talking only one aspect, which is to expand the structural diversity of this kind of systems. This is very important because most of the time when we talk about these structures, we make an analogy with either classical polymers or with biological structures. Okay, so if you really want to make a comparison with classical polymers, we should be able to achieve everything what classical polymers has, not only in terms of its structure, but in terms of structural diversity, multi-component system, block of polymers, and functions. So everything is very important if you want to really compare this system with classical polymers. So this has been our attempt to improve this structural diversity in this case of polymers. That's what probably I'll be talking about today. So the starting point was this. So the mechanistic insight into the supramolecular polymerization process. So a lot of input has been made, especially during the last 1 decade. And the biggest realization is that the mechanisms with which these polymers are grown is very similar to the classical polymers and also very similar to the biological assemblies. So people found that there are 2 types of mechanisms exist in this kind of polymerization growth. One is basically the isodesmic, which is a terminology taken from protein self-assembly. It's an isodesmic model. It has an equal association constant. If you look into the classical polymers, it's similar to the step polymerization. So it grows step by step. And the other one is obviously the cooperative system, which is a nucleation growth. It has a kind of energetically unfavorable nucleation step followed by the elongation. So this is very similar to the chain growth polymerization in classical polymers. And also this is very similar to most of the protein self-assembly works. This is a nucleation growth system. So this realization was very important because the kind of structures we formed highly depend on what kind of mechanisms it follows. Okay, so we know that if it is isodesmic, if you want to get a high molecular weight polymer, we have to really push this self-assembly to almost 100% completion because it's like the final step. These oligomers are combining to make the classic the large molecular weight system. So we have to really push it to completion. This is not easy, right? But in nucleation growth polymerization, at any stages, it's an equilibrium between the long chain polymers and monomers. So you get, even if you don't push the reaction to completion, you have larger structures in the system. So this is well known in classical polymers between step polymerization and chain polymerization. The realization is similar mechanism is followed in this kind of self-assembly, the supramolecular polymerization. So this is a good starting point. So what I'll be, for the time being, I will be not talking about this isodesmic systems. I'll be mostly talking about cooperative system because we would like to make large molecular weight polymers. And always when it comes to self-assembly, it's better to follow this mechanism rather than isodesmic because most often we don't get larger molecular weight systems.
So we just explain to everyone the temperature dependence curves. What do we, why they're different?
Sorry. So this is actually the growth. We follow the growth process. So how do we make these supramolecular polymers? Either you can do it under kinetic conditions or under thermodynamic conditions. So in this graph I mentioned, so you go to monomeric state and you cool down very slowly to ensure they are under thermodynamic control. And we follow this growth process. So traditionally people have been doing it with under thermodynamic control, but recently more we are going into a kind of kinetic conditions similar to the protein self-assembly because that has a better control on the structure. So I'll not be talking about this temperature things. I'll be more talking about the kinetic aspects. So this is the most important slide, which sets up the rest of my talk. So what we found that, so as I mentioned, during the last few years, it's very important to control its structure. That means most of the supramolecular polymers before we made was polydisperse. So what was the key to control its structural characteristics? And there, I mean, like a lot of mechanistic insight has gone into it. And what people found that, let's talk about nucleation growth mechanism. we know that nucleation step is energetically unfavorable, but most of the synthetic system, what happens is the activation energy is very small. So this nucleation is spontaneous. Okay. So if you like to control the monomer molecular weight, so to the polymer molecular weight, it should be a living system. So again, I'm taking analogy from classical polymers. All of you know that what is a living polymer. Living polymer is that if you introduce another monomer, it grows at the end of the chain. Not independent initiation never happens. Okay, so that's very important if you want to get a control on the structure. Okay, so if you consider just a nucleation growth polymerization, what happens if nucleation is extremely fast, if it is spontaneous, what happens if you introduce additional batch of the monomer into the system, it starts its own nucleation. Okay, it will not grow at the end of the polymer chain. Yeah.
How about secondary nucleation?
I'm coming to that. So that's basically the today's talk. Okay, so most of these structures are going to the primary nucleation. Comes to the secondary nucleation is the only way to address the higher hierarchical structures. That's what we have been doing during the last 2 to 3 years, and that basically comes to today's topic. So I just wanted to introduce this primary nucleation, and then we'll talk. So this happens. So the challenge was to slow down this nucleation step. So we have been trying to do it by a kinetic delay in this process. There were a lot of molecular programming has to be done in terms of monomeric structure if you want to control this nucleation process. So once you slow down this nucleation step, what happens is if you introduce additional batch of the monomers, it starts growing at the end of the polymeric chain rather than independent nucleation. So that is why it is called as a living polymer, not from a biological perspective of a living. Its chain ends are always active. It's never dead. So it is growing at the end of the polymeric chain. And this challenge was quite different from classical polymers, because classical polymers, the propagation step was the one to be addressed, because you know that propagation step is extremely fast in a chain polymerization reaction. And once two radical forms, it's quenched and it forms a dead polymer. It can never grow. But that's not a problem in supramolecular systems, because it's a non-covalent association. There is no termination actually. It can grow at any point. So termination is not the issue here, but the issue was to address the nucleation step.
It happens to achieve the supersaturation. Right? It's just that.
Yeah. So the typical, what we see that where is the nucleation happens in most of the systems, we found that it's a very small number, like 3 to 4 monomeric units. It depends on the system.
Oh, just at the 3 to 4.
Yeah. It's still in solution. Everything I'm talking about in solution.
But they are aggregating.
They are aggregating.
Yeah. So then if it is not under supersaturation, then what is the driving force for them to?
So it's most of the synthetic system is enthalpically driven system. So there are hydrogen bonding system which drives the systems to come forward. And once they have a critical size, they start growing.
Yeah. What I'm trying to understand is, so you have a molecule dispersed in the solvent. They're stabilized by the solvation of the solvent molecule. So in general, when you reach a certain concentration, right, then the total energy of the system is.
So we are not playing. Yeah. So this can be induced either by concentration or in terms of like most of the protein assembly with time. So they try to come together with interactions, and then once this reaches typical size, it starts nucleating.
Okay, so it's more like a seeding, and then you grow onto that.
No, seeding. The first is a growth. Then you can always come with an additional monomers. Then there's a seeding. So they're already pre-existing nuclei is there. The first growth never happens in presence of seeding. It just starts growing because there are interactions which monomer comes together and starts growing. So there is no seeding process there. But then once you grow and you come with another batch, it starts seeding process happens. It starts on the existing nuclei.
Okay, thank you.
So this is we call as a living supramolecular polymerization, because if you come with, since the nucleation step is kinetically delayed, it will not start its own centers. It will start growing on the existing chain. So this is good. So how people addressed, how people achieve this kinetic delay is by a molecular programming, which I'm not going through it. But today morning also there were wonderful talk about off pathway nucleations and on pathway nucleations. So what in synthetic system, what we have tried is to trap these monomers in a kind of metastable phase, which most of the time is a kind of off nucleation pathway. Or there is a beautiful system from IDA where conformationally folding these monomers to create a kind of kinetic delay. And very recently, what we are trying to do is, like Kote talked about today morning, is to use like a kind of liquid phase separation as a kind of metastable phase where we can create a kind of kinetic delay before these polymers are growing. So there are many approaches. We have also contributed to many aspects into this. So this is well understood. Now we can modulate the monomer structures and thereby introducing a kinetic delay in the growth of these assemblies. So what is next? We also, I'm just mentioning this part from our group because this is very important in a later stage of my talk. That's what I'm just mentioning this right now. We also came up with a very similar to the biological structures. Biological structures, the growth happens is a kind of reaction coupled, where it is connected through a kind of reaction, and the rate of the reaction determines the growth of, for example, ATP binding or whatever. So we try to mimic that in synthetic system. So we are trying to make also the growth of these chains coupled to a reaction. Okay, so we have used enzymatic reactions, we have used redox reactions, we have used photoredox reactions where the rate of the reactions which control the growth. So that is very similar to the biological assembly process where always coupled to a reaction. The beauty of this kind of approach is, if you look into this, you need a lot of monomer modifications. Okay, that makes the predictive design a difficult problem. Every system should have a different design. So we thought that probably if we can control into a kind of reaction coupled, it can be a kind of generalized approach. And for chemists, there are many opportunities when it comes to a kind of reaction control systems. So we have shown it with various systems. And the beauty of this kind of system is that it can be modulated completely by the reaction conditions. So this is something which we have shown. And we have one of the well-established system is the kind of enzymatic reactions. So we have made monomers which are like a kind of ATP sensitive, ATP selective. So you can do an in situ ATP synthesis, and the rate of that ATP synthesis controls the reaction growth. This is something which I will be talking towards the end of my talk. So this is all about primary structures. Since time is running out, I'll just quickly go to today's talk. So once we have this kind of living supramolecular polymerization, what we have achieved is we can get structures which has a good dispersity, close to like 1.1, 1.2, which is a very good numbers in kind of this kind of structures, and also control the molecular weight, because once you have a structure, you can come with the monomer, and monomer to the seed ratio determines basically the molecular weight. So if you want a 10,000 molecular weight, you take the seed to monomer ratio, which determines, since everything is living, because it doesn't start its own nucleation centers. So that way you can control the molecular weight. These are the couple of things which we have achieved during this polymerization. We have also, next level of primary structure control was to go into the multi-component system. Can we grow like a kind of two monomeric systems and make block of polymers? This is very important because whenever we make a comparison with classical polymers, one of the questions often comes to us is, can we make the kind of structural diversity classical polymers has? And you know that block of polymers are important in classical polymers when it comes to lithography or in terms of photovoltaics. So can we make similar structures in the classical polymer, in the supramolecular polymers? Since we have living conditions, what we can do is we can think about another monomer, which is structurally similar probably, but different optical and electronic properties, since most of the monomers we work has a kind of optical and electronic functionality, so that it also a kind of bottom-up approach to make nanostructures, which are functional nanostructures. So we can come up with this, it starts growing at the end. So these are all completely under kinetic control, and we can make heterostructures like inorganic heterostructures if you carefully choose these monomers, which is either optically or electronic complementarity. So that way we can make structures which are heterostructures, which are very similar to the inorganic heterostructures. So these are also is very easy once the living supramolecular polymerization is done. So just I'll skip this part. Believe me, we are able to make structures which are like this block of polymers, which is made up of two components. So you can see that we work with molecular structures like this, pi-conjugated systems, as I mentioned. Since we are organic materials groups, our monomers are like this kind of pi-conjugated systems. And one of the well-known class of molecules we studied is naphthalene diamides. The beauty of the naphthalene diamides is obviously we know that it's a kind of N-type semiconductor. But the beauty is that just by some small changes in the core, we can change this optical and electronic properties over a kind of wide range. So that's very important for us when we want to make functional nanostructures. Okay, so that's why we stick to naphthalene diamides. And you can see that we can extend to these two component systems, and we can make really blocky supramolecular structures very similar to the classical block copolymers. And the red component is an energy acceptor, and the green component is an energy donor. So we can really look into make structures like this, look into the energy migration, and Arindam is looking to some of these structures. I quickly gone through it just to say that these are the one-dimensional supramolecular polymers we are talking about. We are in a kind of fairly good control in its primary structures, both in terms of dispersity, its length control, and probably a kind of two component systems where we can go to make heterostructures. Okay. We can increase the complexity to tri-component. This also we have done, and all on kinetic control. So that brings to today's talk. Yeah.
So your heterostructure, what is the driving force? You have a functional group, like why they are alternating or something like that?
Yeah. So so that means I have to go back to this slide.
No, maybe you can then delay.
No, it's okay. It's okay. So so there are two ways. One, we can make this when you take two monomers. If you can completely make these structures under thermodynamic control, then it completely depends on the reactivity ratios between the free energy of interactions between the two monomers. So that way also we could make it, but that is challenging because it's not easy to predict that free energy of interactions between the monomers. So what the most, we have started, and many others have now last 2 years, there are many reports came up. So what we do is under kinetic control. Okay, kinetic control, the same way living supramolecular polymerization I mentioned. So what we do is we make a seed of one of the structures and put this monomer into a deep kinetic trap, second monomer, so that it doesn't start its own nucleation. And then you introduce in presence of seed and it grows at the living ends. So this happens only if it is a kind of living seeded supramolecular polymerization. Otherwise, you will completely end up in a self-sorted structures. One is growing and it has independent nucleation. So this can be done only if you kinetically delay the nucleation of the second monomer. So that we do it by our molecular programming. So that way you can make these structures very similar to the classical inorganic heterostructures. Okay. So
can I just
just
maybe a quick question? The part of the design strategy here is essentially disallowing the mixing before. So you just have one particular component in solution, you let that grow, but there will be some heterogeneity in that seed.
Yeah, so seed heterogeneity is an important thing. What we do is we make the sonication of the seeds, and then we make them monodisperse seeds. That's very important.
So that is the starting point is very critical, right?
Yeah.
Then you do the living sort of edge, both the edges.
That's the way we do.
And then again you stop. Again you reintroduce the original seed.
That's the only way. Sequential addition is the only way we can make these kind of structures.
Okay, so it is like in some sense covalent synthesis, but with multiple steps.
Okay, but then ideal situation is doing under thermodynamic conditions. In classical, if you look into classical block of polymers, there are two ways they make. One is by ATRP. That is also under kinetic control. You make the microinitiator and add the monomer sequentially. But there is another method by chain copolymerization, which completely depends on the reactivity ratio between the monomers. So if you have the reactivity ratio is just above 1, you will be getting a blocky system. This is exactly why chain copolymerization has been used to in conventional
Very nice. Okay, got it.
But to do it in supramolecular system, we have done it, but we have only one system. And so far, only one more group reported based on the reactivity. Then it's stable once you make under thermodynamic. But it's not easy. That's why people are more moving to the kinetic control and making it. So far, we could get the primary structure. And as I mentioned, we are the people working on also on pi-conjugated system. So if you look into the heterostructures, very similar to the inorganic heterostructures. That's why we are able to look into systems like this, and probably you can use them for energy harvesting, photocatalysis, and so on. So this is something which we have to come up with many more systems. But then that brings Sudipta's question, and then what is next? So next grand challenge we feel is, and this is often the question that comes to us is, if you want to always make a comparison with classical polymers, I should be able to make branched systems. I should be able to make star copolymers. graph copolymers, how do you do for a supramolecular chemist move away from one-dimensional structures to two? So it's not only, it's important for addressing the structural diversity. To expand the structural diversity of these supramolecular polymers, we should start addressing the other dimensions as well. And also, if you are as an organic material chemist also, it's very important if you want to make structures very similar to the other kind of heterostructures like layered or kind of core shell or something. It's not, we cannot deal with only primary nucleations. We have to come up with alternative strategies. So that's why today's talk, I'm trying to try to control the molecular programming at the higher hierarchical level is possible. And that's the only way if we want to expand the material functions as well as structural complexity of this kind of systems. So how to do it? Again, as mentioned, we always get inspiration from biology. And in biological structures, there are, as Sudip rightly mentioned, not only the primary nucleation, which is actin self-assembly, if you look into it, beautifully follows primary nucleation. But as soon as it goes to amyloids kind of systems, secondary nucleation sets in where the surface of the existing seed acts as a catalyzing for the incoming monomers to grow. Okay, and that is in fact one of the major factor for we can kind of catalysis of the amyloid fibers. So, and for a supramolecular chemist point of view, that's the only way also if you want to address the higher hierarchical structure, you want to tap the secondary nucleation. But the question is, we don't want to do it in a random way. We want to really do these things in a kind of precision manner. Can we control the primary nucleation events and secondary nucleation events by a molecular level control, and probably can be used it to form the branch structures? This was the this is the motivation, and I will be showing that our recent results, half of them are not published. So I will be really happy to get your feedback. And again, thanks to the protein self-assembly, all the kinetic models are well established how to distinguish between the primary nucleation and secondary nucleation. So one of the well-known method is to look into so we'll be talking only about the kinetically controlled growth in this case. I'm not talking about the thermodynamic conditions at all. So what happens is, so if you put the T50, they have the T50 along the amylase with the monomer concentrations, and that scaling exponential can tell you whether it is a primary nucleation or secondary nucleation. The beautiful papers from Knowles, and there are a series of papers from Knowles on the protein self-assembly. So what happens is the scaling exponential, if it's less than 1, it generally or close to 1, it is a kind of primary nucleation. And if it is more than it's a kind of higher order structure or secondary nucleation can happen.
So quickly, could you define T50?
T50 is the time requested to reach the half of the monomers. It's a degree of polymerization is 0.5. Okay. So also there are models available, and if you do the seeding, so like the question before, so if you have a seed, and if it is primary nucleation, you see the spontaneous nucleation. You can see that it grows immediately. And if it's secondary nucleation, only what most of the time we see is a decrease in the lag phase. This is a lag phase, decrease in lag phase, but still it grows. Okay, and then this can be fitted into the models, primary nucleation models, secondary nucleation models. We can get it. I'm not a real expert on the mathematical models, but we can do that, and we can distinguish. My message is that we can easily distinguish between the primary and secondary nucleation events, provided you have a beautiful kinetic curves like this, where every process, every stage of your growth process is you have enough data points. Otherwise, we can be in trouble. So their importance comes to our spectroscopic probing. So since we work on pi-conjugated monomers, and they're very sensitive to the intermolecular interactions. So we are able to get beautiful kinetic profiles at kind of many data points because we are able to in situ probe the structures. That's the key, because if instead of spectroscopic probing anything else, it's not easy to fit into this primary and secondary nucleation models. Okay, so this all started with Savi a couple of years back.
So even before you talk about secondary nucleation, doesn't secondary structure, can it not form? Because your structures are long enough, right? They can fold and form all sorts of...
That is fine. So the topology of that long polymers, the persistent length and everything, which I'm not talking, that's not the way it is indeed possible. Okay. So what my point is that if you want to make a branches from a backbone to make a graph polymers, so we have to really tap this secondary nucleation. That's the only point. So a primary polymer, further changes in topology is not something which I would like to address. I'm like, that's not the way.
But wouldn't it kind of mess with your results, or whether you are seeing secondary nucleation or just secondary structure?
I agree, but most of the systems we work has a very good persistent length.
I see.
Okay, and that's why we don't really a kind of random coil structures or something. We don't get into that. Okay, so we have a very kind of rigid structures in most of the systems we work on. I agree completely to you. There are people like a Shiki from Chiba University. They are really looking into how the folding process of the supramolecular polymers is working.
Yeah.
So that is a kind of different aspect.
Yeah,
that's
especially
not
because you have non-covalent interactions holding them together. That's why I thought they can, be crazy.
They can be crazy, but the kind of systems you can see, you would be seeing some of the images. We have a very good kind of persistent length, and we are safe in that way.
Okay.
We don't really address into that
Okay.
kind
And
of
that's
system.
because the molecules chosen and the
By
way you are designed.
Yeah, that's
Okay, thank you.
Yeah. And also, I'll come back to this probably towards the end of this talk. Just to this all started like during that COVID time when we are reading many things. We got a lot of time to read. The Souvik this started with a very simple observations in our group. So Souvik was working on this kind of very simple organic systems. So this is if you look into this molecular design, only two aspects is important. It has a strong chiral core, binaphthyl core. The beauty of this binaphthyl core is as a kind of, it has a strong homochiral interactions. It's used extensively in asymmetric catalysis and so on. So if you have an amide group here, always it has the same enantiomer only will grow on top of it. Okay, the opposite enantiomers will not grow because it has a strong opposite homochiral interactions. That is very important because that is the chirality is the key we are trying to manipulate primary and secondary. So this is homochiral interactions of this binaphthyl group is very important. Okay, keep it in mind. In the next slide, it will be more clear. So we have a binaphthyl kind of this amide group here. And also this is a pi-conjugated system we are talking about it. This is an naphthalene diamide. And the beauty of this system is once it starts interacting with each other, it has beautiful spectroscopic changes, which we can selectively monitor and get kinetic profiles like this. Okay, so either with a circular dichroism, because if you probe the naphthalene diamide, this chirality will induce circular dichroism, and we can probe the circular dichroism of the system or fluorescence or the absorption changes. Okay, so there are parallel spectroscopic changes we'll be able to monitor, and we can get kinetic profiles like this. Yeah.
So you start with chirally pure monomers, or you have growth which has some strands which are left and other are right.
We do both, but so in these cases right now I'm talking about enantiomerically pure molecules. We starts growing. The whole idea was to do a resolution, but then that turned out to be, I'll come. So this beauty of this system is, it is a kind of, you see that there is a long lag phase and there is a growth process. So it is a kind of kinetically controlled system. The nucleation is not spontaneous. That's the only point. It has a delay. And that delay is because this molecule can form kind of intramolecular hydrogen bonds. So that is the kinetic trap state, and which opens up and then starts growing. And that's the way it is. That information, we got it from MD. We don't have an NMR proof or anything, but molecular dynamic simulations with Bala Subramanian's group clearly suggest that this kind of the dormant state of this monomer is a kind of intramolecular hydrogen bonded state. So it's growing and it has a living nature. You can see that if you introduce additional monomers, it starts spontaneously growing. It's a primary nucleation. It starts growing. You can do this multiple step. You look into that, length also grows in a kind of living manner. This straight line tells that, and dispersity all through the seeding process remains constant. So these are all the characteristic of a living polymer. Okay, so this is all from this monomer. But what is interesting is the next slide. So what we try to do is a kind of seeding process here. The whole idea was that if you take a Resovic mixture, as Sudipta mentioned, if you introduce one homochiral seed into it, can you do a kind of Louis Pasteur's kind of resolution? Okay, this was a simple concept. We try to do it because it has a strong homochiral recognition. So what we have found is, yes, if you add the same monomer, this is the lag phase immediately decreases and it starts growing spontaneously. That is the primary nucleation. But opposite enantiomer, when you add it, you see that it did not grow spontaneously. Okay, but what has happened? It still grows with a reduction in lag phase. So this kills the concept of a resolution. It will never work because the second monomer also starts growing at some point. It will never work. So this is what we try to investigate. The only possibility that since it has a strong homochiral recognition, the opposite enantiomer will not be able to grow on the primary surface because that chirality mismatch will never allow to grow on the primary chain. So the only point is probably it is growing under on the surface of and it could be a secondary nucleation. That was the whole thought process. So we did detailed kinetic analysis. We varied the monomer concentrations, look into the scaling exponential and everything. And then very, very clearly, we got an indication that one is very primary nucleation kind of things. And if the homochiral seeding follows a primary nucleation and heterochiral seeding follows a kind of surface catalyzed secondary nucleation. Okay. So we have clear proof from the spectroscopic data, kinetic data. Okay. So this is summarized here. So if you take a seed, take the same enantiomer, it grows at the end. If you take the opposite enantiomer, it grows on the surface. But then the disappointing fact that we were really excited to go back and check the microscopy. Okay. So, but then we did not see anything. We did not see any branch structures. Okay. And that is always also true with amyloids. Yeah.
Your surface chirality is different than
no. So surface chirality has nothing to do. Okay, so just keep concentrating on molecular chirality.
No, why I'm saying this is because you said one enantiomer follow one path onto the surface.
Yeah.
Okay, so that's what
the chirality, the surface chirality is not triggering anything. What happens is if you add an opposite enantiomer, it cannot grow at the chain end because there is a strong chirality mismatch. But it will still look for a seed for heterogeneous nucleation, and that is the surface of the fiber.
No, so the surface that I'm referring to is not this, but this portion, right?
Yeah,
so
you are talking about. So I have a cylinder.
Yeah.
Which part are you talking about it?
This part.
So that has a chirality. That's why the only same enantiomer can grow there. Opposite enantiomer cannot grow there. That is driven by the chirality. But then if the opposite enantiomer, since it cannot grow there, it will look for a surface. Because it's a kind of, it will try to look for the kind of easiest path to get nucleation. And that is a surface. And I'm telling that surface chirality is not playing a role because we have done some control experiments with an achiral monomer. It doesn't have whatever growing on the surface does not have a chirality. So that's what I'm saying. So we have ruled out surface chirality, any effects on the surface chirality. Yeah. Is it clear? Yeah. So, what we expected is some branching to happen on these structures. We thought probably that's the best way to make branch structures, secondary nucleation. But then we realized that it happens on the surface, but it detaches from the surface. Okay, that's well known in amyloids as well. So it starts growing on the secondary nucleation surface and it detaches. So this will not provide you a way. This is nice. We have spectroscopically probed it, but this will not be the solution for me to make the branch structure. So what is next? So it is very clear. Again, today's many of the talks in the morning was very useful. We need to come up with some sticky side chains so that even if your secondary nucleation happens, it should stick to the primary fibril, parent fibril, so that we would be able to stabilize the secondary structure. So that's the only way we would be able to use, if you want to use this as a kind of secondary nucleation as a method to stabilize the higher order structures of supramolecular polymers, we need to come up with some sticky side chains. So that was the lesson we learned from here. So we were looking for structures. We screened a few structures. And I think we really got interested because this has the potential and this is probably the only way we can make branch structures. So this Souvik again came up with some interesting system. So keep it in mind. So chirality, so we need a mismatch. Okay, so what we have shown now is even probably biological systems will not have that control. So we have a molecular level chirality control to tell the molecule to go either primary nucleation or secondary nucleation, because that chirality mismatch biases the pathway of primary or secondary nucleation. Okay, so with that in mind, so we came up with peptidic designs, which is very similar to the beta sheet structures. And again, you see that this is one enantiomer and this is opposite enantiomer. And this rules, it's not only that one system we found, most of the system we make we are seeing it. I think people have observed in the past, they did not look into the detail how this kinetics happens. Okay, probably that's what I feel. I mean, people miss it. So what we found that the same rules follows. If you do the homochiral seeding, it forms the primary nucleation. If you do the heterochiral seeding, it forms the secondary nucleation. Okay, fine. But then what is interesting was that when you look into the from here and TM and many other things, you see that now the secondary structures formed is sticking to the primary fiber. So that sticky change from the hydrogen bonding sites is really helping for the next growing polymer to stick to the surface. And how do you confirm is indeed the secondary nucleation? You see that you vary the enantiomeric excess, you change the length of the secondary fiber. Basically, that determines how much it should grow. I should admit the fact that I don't have a control where it exactly nucleate, okay, because that surface is always available. I will not be able to tell the molecule right now. It has to be at the end. Obviously, we have to work towards that. We have some ideas and we are working towards that. So you see that sometimes you see it at the end, sometimes you see at the middle. But then overall length of the secondary supramolecular polymers is directly proportional to the enantiomeric excess of the systems, and we work for it. So these are the secondary supramolecular polymers.
Can I ask a quick question? Yeah. Those sites where you have this white color, right?
Yeah, on that two fibers.
So there are two fibers, right? So
either you see it in the height image or in the face image.
Right. I believe that. What I want to know is the molecular reason for that. What is the interaction?
So this is right now, because we don't have a direct way, because we are trying to probe the presence of hydrogen bonding by urea concentration. What happens in the primary structure?
But you kept it cleverly in the design somewhere, right?
Exactly. So that is we believe it's a hydrogen bonding site, because only there are many control molecules we made, we could not stabilize it. Okay. So right now the MDs are going on to get a better idea, but right now I don't have but what we have proved only with controlled molecules, both sides peptides, one side alkyl, both side alkyl, and we strongly believe that it's hydrogen bonding sites which stabilizes the secondary structures. And this was not enough. We wanted to prove more about this is indeed true and it is possible with a wide variety of systems. What we have done is Saikath, who is continuing this work right now. So what he has done is he has modified. So you can see that the previous structures is only one substitution. So we will not be able to distinguish between differentiate them from a kind of microscopy. Fluorescence optical microscopy is not possible. So what he has done is he slightly changed the substitution at the core. So one is oxygen and one is amine, isopropyl amine. And you can see the color from green to red. And these are the, and again, yes, you see that all the rules same. It beautifully works. Homochiral seeding goes to the primary, heterochiral seeding goes to the secondary, and it works. And you see that these are the corresponding TM, AFM, and this is a structured elimination microscopy images where you can clearly see that two fibers are indeed sticking, which is also opposite and opposite colors. Okay, so this is really working. The secondary supramolecular polymerization that way is working, and they are able to stabilize.
And it's a quick question. Why do you always show in the cartoon it's exactly stacked like this, and it can't have this slip? A bit slip, then again a slip.
Agree. Slip. Yeah. So it's possible. So what we have done is, so this we don't have a control right now. What happened is that one of the systems we started this here and started seeding. This forms longer polymers. Okay. So we have reversed it. So then the other one will come. It depends, because you see that now it is two different monomeric systems, and the degree of polymerization, molecular weight is slightly different for that. That's why it happens. It can always stick.
The reason I say that is because you might have exposed, because the slips on the one array, you might have exposed some hydrogen bonding which could be picked up.
Could be. Yeah, JD, I completely agree. I'm like, we need to.
Maybe the structural.
Yeah, so this is one of the toughest questions we had to face in publishing this paper, because we have to really put it the molecular level interactions. We are well aware of that. So we are working towards to come up with the systems to clearly know what is really triggering that molecular level interaction. So we are now getting more and more confidence that it is working with the diverse systems. And so we are indeed able to use secondary nucleation to stabilize the secondary supramolecular systems. Okay. And No.
When the structures that you see, they are not rotated up even a bit.
Yeah, there is a rotation.
That would be there.
That would be there. But then what happens is that there is no complete helix is forming because to get a CD signal, there can be dimers or trimers. The other one. So what is that? That is controlled by the chirality. That's not it.
Sorry, quickly, I guess, do you have something like a defect on these single chains?
Because the defect sites could be the triggering points.
Right. So in that case, did you say do some sort of temperature dependent study? Because you could assume that defects would be temperature dependent, possibly in this case.
Yeah. So that's a good suggestions. We also thought about it. The defects could be the starting point of nucleation that we have to keep studying. We have not investigated that aspect. And also one thing I just want to mention, we have very weak interactions because when you start sonicating, this guys comes out. Yeah, the primary structure is intact, but then these structures are coming out.
Nice. Very nice thing. See this, you are using it in a non-polar solvent, right?
This one is non-polar pot solvent. I have aqueous solvents also coming out.
Yeah. So especially in case of aqueous solvents, can water participate in these interactions?
Yeah, solvents are crucial. Got more and more now. What we learned from the systems is that solvents is very crucial in determining the landscape of this system.
Because in case of protein structure formation, water has a very important role.
Yeah.
So in this case, solvent, you have not found any.
No. So the only point is that we don't have a flexibility to vary the solvent because every system works in a particular solvent composition and so on. So we don't have that luxury to study that solvent effects, but I agree completely to that solvent has a big role to play. And especially we do see it in aqueous systems and we have to. If you're not sure about what interactions are causing, right now we are sure about it's a hydrogen bonding interactions because that's key. Because any other cases we made a lot of control molecules, they are not sticky, they're coming out. So we are sure that there is a specific interactions like hydrogen bonding which is stabilizers, but that could be like JD mentioned, something is pulling out. That much I'm sure about it. Is a hydrogen, because we don't see this stabilization of the secondary fibril in any other cases, all the control molecules we have seen.
So does that guide you in selecting what kind of substituents that you should put in to lengthen this chain and...
So lengthen chain is always not a problem because what we can do right now, we have done it. So we can, if you want to lengthen this, I can do the seeding here with the primary. If I added extra monomers here, it will start growing here. So that's possible. So we can manipulate slightly different way the length of the systems by seeding process. Primary nucleation can be done. We have done it. So take these structures, we add additional monomers. If you add red monomer, it will grow here. If you add yellow monomer, it grows there. Those things, manipulation of the length is not a problem for us. We are just primary structure, we are under control now. Don't have control is this.
So I'm kinetically regulating the questions also.
When you mentioned that before in case of using non-sticky monomer, so you told that those are not stable. They suddenly comes apart.
They nucleate suddenly or not, but by the time we look into the microscopy, they are out.
So any time scale or level of polymerization, at what level it comes out?
Very difficult to probe that time scales, but then because spectroscopy does not give me any manner to look at that time scale. I don't know. It should be Q. I have a feeling that because when I look into some of the beautiful amyloid papers, what I see is that they nucleate, they come. That is the way autocatalysis work. And that guy again nucleates. Okay. So that's the way autoproliferation happens in amyloids. So the secondary fibers happens, that acts as another one. So that's the way it works. But I don't have the time scales with me. I don't know how to probe it, that time scale.
Also, like in case of non-sticky polymerization or sticky, is this possible to cross-link to chains by photocatalysis?
It's possible. There's another. So what we are, instead of kind of cross-linking you are talking about, we are coming with a kind of bifunctional monomers. Okay, so that's something which we are also doing, but slightly different system in a charge transfer system where we have two components, donor and acceptor. So that acceptors we are making bifunctional, so that the kind of physical cross-links we are purposefully introducing, we are doing it.
Okay, we will continue with the talk right now.
Yeah.
So you have a set of statements that you can make for the talk.
Yeah. So these are the more things, and maybe this can expose. I just want to show this slide to say that again, to just convince that it's not really one system. Okay, so again, this chromophoric system with a strong chiral recognition, Darshana, which is working. So what we are, so shish kebab I put it because in polymers, classical polymers, shish kebab is one of the well-known secondary structures because it's grows and then it forms a kind of branches into it. So again, we see that is indeed possible. You see that enantiomeric excess, as we keep increasing in this particular case, the branching increases. So just a preliminary data we are just, so it happens and we have beautiful spectroscopic data. Again, it happens, homochiral goes to primary seeding, heterochiral. So the chirality mismatch is giving me a control in this case also. So it's many systems, it's uniform. Okay, so it's a branching we will be able to control by the secondary nucleation. Now, that's okay, but then can we do it in a real precise manner? So this is a structure. If you look into classical polymers, people call it as a graft copolymer. Okay, so graft copolymer is slightly different from the secondary nucleation in the sense these monomers are different. These are backbones are kind of different monomers. So these two monomers are different. And if you look into basically so the next, obviously I have some results, otherwise I would not have shown these schematics. So if you look into, if you want to make such structures, obviously now you got an idea how we are going to do it. We are going to take the primary backbone and come with an additional monomers and try to secondary nucleation in presence of that primary backbone. That's the whole idea. So now there are two, so I'm just switching from non-polar solvents to aqueous solvents also, because as I mentioned, I'll be using an enzymatic control reaction to do this. So these two are important. So you need to bias the primary and secondary nucleation. So you need a tool. And the previous case, it was chirality. And if not chirality, what else? And one of the things we are trying to do is a kind of molecular organization. So in pi-conjugated system, there are two types of organization which is well known. One is a slip stack and one is cofacial. And one is called as J aggregate and the other one is H aggregate. And we think that even if you have such a difference in molecular organization, that could be the one which prevents the mixing of two different monomers. So what we need is one monomer should not go and sit on the other monomer. In the previous case, it was chirality. I hope I'm not confusing. And in this case, it is the molecular organization, which I'm going to talk about it. Okay. So how to do it? So this is a bottle brush polymer. So the bottle brush is again in polymer is a densely grafted polymer, because if each monomer sequence, if it has a side chain growing, you call it as that kind of graft copolymer is called as a bottle brush, because it's a heavily dense polymer. And also from an organic material point of view, it would be nice. You can think about it as a kind of energy harvesting guys, harvesting to the backbone, probably collecting it for catalysis or something like that. It's also possible. I'm like just to add some flavor to that. So if you look into, there are Machersky is one of the first one to make this kind of structures in classical polymers. There are many ways: grafting to, grafting through, grafting from. But then for a supramolecular chemist, probably the best way is to use a grafting from. You have a micro initiator. My micro initiator will be the backbone, and use that as a surface catalyzed center initiator to grow these monomers on the side. So that I'm going to show is a grafting from approach to make these kind of structures. So the Anshuman is working on it. It was a kind of very long, last 2 years, his research I'm going to summarize in like 3-4 slides. So what he has done is he came up with these kind of systems. Again, naphthalene diamides. So it has two for substitution. One is isopropyl amine and other one is a thiol. So one is a kind of green and one is red. So when they aggregate, there are two different colors. The whole idea is that we are able to monitor and we will be able to differentiate between these two monomers. And you see that it has a ganidinium site. And ganidinium site, we know that ganidinium can bind nicely to phosphates. Well-known sensors are known. So this can bind to ATP and trigger polymerization. Okay. So a bit complicated, but so two levels of controls we have made to make sure this brush forms. One is we made them with two different synthetic pathways, and it has two different molecular organization. So that only condition is that they don't mix. They are forming separately, and we have two different synthetic pathways to make it. And the backbone, we made it by a kind of kinetically control, like putting them into metastable state, and then grow and form the fiber. And the other one, the brushes, we made a kind of enzymatic approach. So the whole idea is, if you have a backbone, you use an enzymatic approach to in situ grow the second monomer. So it starts growing on the surface. Okay. So just to cut the long story short, so we have the first one. This molecules forms one-dimensional fibers by a kind of pathway complexity. It is an off-nucleation pathway. I'm not going to the details. So what happens is, for the brushes, we have used this molecule. So what happens is, this is highly ATP selective. So what we do is, we start with ADP, use an enzyme, creatine phosphokinase, in situ generate ATP. ATP binds to this molecule and it starts growing. Okay, so it follows a kind of nucleation growth. And the beauty is that this growth can be controlled either by the concentration of the ADP, concentration of the enzyme, or concentration of the primary fuel, which is phosphocreatine. So we can play around with to control the growth process. Okay, so that's the beauty of a reaction coupled polymers, because we can use any of this any of these parameters one can change, and we can make this growth process. So what we have done is, we get structures, growth structures, which are quite polydispersed. So the monodispersed. And the most important experiment is the next slide. So what we have done is, we have taken the backbone. So, and then in situ start making this molecule by an enzymatic reactions. You can see that then from the kinetics curves, it's very clear that it goes to a secondary nucleation. Okay. So now the seeing is believing. So the next slide tells you this is the kind of bottle brush polymers we are having right now. So this is a structure elimination microscopic picture. So this is the backbone, and this is the brushes which comes out of it. One is a H aggregate, one is a J aggregate. So backbone is an H aggregate, but we have colored with an additional dye to make it visualized in this. And this one is, the brushes is a J aggregate. So it varies, the backbone thickness or the brush. The backbone is quite thick. They are quite firm. There is you can see that the brush length, we are changing it. I'll come to the next two slides. We have a precise control on backbone length, brush length, and the grafting density.
The backbone itself is 1 micron or so.
Yeah. Backbone is quite thick. So this is something which is, we are trying to sonicate it to make it, but then they are only making it in the shorter direction, not it. So that is not a kind of single fiber, single column. It's not a single column. That's my point. There's many molecules. Yeah. So, and also these are the TM images. So this is a elemental mapping. You can see that there are two different monomers coming. So as I mentioned, we wanted to change the grafting density. So what we have done is, we have changed the monomer to the seed ratio. That means backbone to the brush ratio. And you can see that fairly, you can control the density at the backbone. Okay. And as I mentioned, we can also change the brush length. So what you do is that you just keep making each sequential addition, add the second monomer each time, and enzymatically include. And you can see that the brush length is gradually increasing. So you do sequential seeding at the brush because it's a primary nucleation. You start growing on the brush. So you can get the brushes and as well. So this is the way we are able to do the secondary nucleation now to make the brushes as well. Okay. How much time, JD, I have?
Yeah, maybe it's maybe 5 minutes. Yeah.
5 minutes. Okay. So my point is that the secondary nucleation which started, like, we are able to now push it to many directions. So we are able to make bottle brush as well. So the whole idea is that what we need is a sequential release of monomers. So we need a backbone, and that surface should be available for the subsequent monomers to grow. This also, now we are thinking how to do it many more ways. So one of the ways Cycap is looking into it. These naphthalene dimers, we have shown that it can form charge transfer complexation with solvents. Yeah, I was looking for it, but no, not for him. Yeah. So what happens is, so if you have two different solvents which have a selective interactions with the monomers, okay, one can, and think that this is a monomer reservoirs. We can think about in energy landscapes, two different monomer reservoirs. The first one releases it, form the backbone. Second one comes and then it starts growing on the surface. So this is something which we are doing because when naphthalene dimers can form charge transfer complexation with aromatic solvents, we can change the aromatic solvents donor strength. So we can have a sequential release of the monomers into the systems. We do see this kind of secondary structure. So this is really working right now. So what we need is either a kind of chirality mismatch or a selective sequential release of the monomers. We are looking into various aspects. One last thing, since JD told 2 minutes, 5 minutes. So this is something which we are trying to do with Uday. So he has been working on this kind of gel systems, bile acid-based gel systems. It's a hydrogels, it's in water. So what he has been doing, one of the uniqueness of this kind of gels is this is a kind of kinetically controlled gelation because he has a metastable phase, which is spherical particles. With time, it goes to this kind of, almost all the metallocolloids is showing similar properties. So it has a metastable phase, which goes into this kind of fiber state. So what we thought, Raju was a PhD student in Uday's groups, and then he joined me as a postdoc. Then we thought, because any hydrogels, the mechanical strength depends on the physical cross-linking and the network topology. Okay, so probably we thought secondary nucleation is the only way. Secondary nucleation can be used as a way to manipulate this cross-linking process because the fiber starts growing on the surface, and that would be able to control the gel strength. And that's exactly what we have seen, that we can change the secondary nucleation links by a seeding process, and we would be able to change these kind of things. So not only from a structural diversity, from a controlling the functional point, also we would be able to change this, use this secondary nucleation. So this group is exploring a wide variety of systems now on this kind of secondary nucleations because he has different metal colloids. He can come up with mix and match and much more efficient systems right now he has compared to this particular system. So this secondary nucleation really works in that way in controlling the mechanical strength as well. So what I've been trying to do so far is to tell you, we started with the primary nucleation to control the primary structures in terms of his dispersity, in terms of his molecular weight and so on. Now we are exploring the surface catalyzed nucleation, highly motivated from the biological structures. We are able to make now branch structures and so on. So when we started, this is one of the science issues, what we don't know. 25 issues were published in like 2008. So 2005. So one of the things is that we really need to push the self-assembly to have more complexity, precision, structural diversity. I think we are trying to push that slowly. I mean, we are learning in this process. One of the important aspects in all this designs is a kind of predictive design, because we are able to work on this particular systems, but it's always an open question when it comes a completely different system. The energy landscapes are different. Its solvent dependence is different. So one has to standardize probably to get it. This is always a challenge in this kind of this supramolecular polymeric field. With that, I would like to stop. Collaborators Bala is extensively looking into the MD. Sarit does the structure elimination microscopic images. Professor Uday Maitra, we are continuing our collaboration on various hydrogels. Arindam is really looking into the exciton transport on these heterostructures. So he has many systems in his hand. He's looking into this. And this field is Sauvik and Anshuman really push this. These are the two guys, and now Saikat is continuing some of this work. Thanks to the Suprachem group. We, as JD mentioned, we just finished 15 years, and thanks to all of their hard work, so that we could really push this field forward. Thank you for your attention, and happy to answer the question.
Thank you, Suvi. Questions? Subi, I have questions, but I will let the audience first ask. Yeah. So, what is the longest polymer you have observed, and at what temperature they are?
So most of the today's talk, we do it at room temperature because 25 degree, we set the temperature and do this. And the length, we can always now, since we have a living polymerization, we can keep seeding and grow it. But at some point of time, keep it in mind that they will crush out of solution. They are no longer in solution, right? That's a problem. So that's probably the limiting factor we are having. So few micrometers, we can easily grow.
Okay.
with the temperature, are you expecting like DNA at particular temperature?
It melts. Yeah. So this kind of kinetically controlled only last 3 to 4 years we started working on it, and before that everything was under thermodynamic system. The same cooperative curves, melting curves, we get it if you heat it exactly similar. We calculate the TM. We do that.
Nice talk, sir. Thank you.
Thank you.
Questions?
Yeah. So one question is, see this backbone when you have almost a micron thickness.
Yeah. Only the last case.
But then the previous description of this is stacking of these one-dimensional system. Then how do we understand the thickness?
Yeah. So this thickness is a question. That's why this paper is not yet published. So the point is that these are amphiphilic systems, ball amphiphilic system. So they are known to form tapes. Okay, so this amphiphilic system. So we believe that these are the dimensions. So ideally, what we like to get it a single monomer. I agree, but we are not able to control this thing. But this kind of packing in these kind of systems are known. These are ball amphiphiles. They form kind of sheets and then grows one DSM system. This is well known in this kind of ball amphiphilic system. And we face this problem as soon as we comes to the water, because hydrophobic interactions are so strong. And they try to grow, stack in all directions because they are typical ball amphiphile. And then they start growing.
Another thing is what is a typical expected response of these polymers to the external fields like electric field and
so this studied. So both under magnetic field and electric field, people are trying to align it. One of the great work from which was in Nature Chemistry from a group in Strasbourg. So what we are trying to do is, because some of the systems we work is also kind of pi-conjugated systems. So how to align between the electrode was one of the biggest challenge. And what they try to do is this kinetic controlled nucleation. They try to do between the electrodes and then align using electric field. Indeed, it's possible. And they bridge the gap between the electrodes and all of them are aligned. So they realize that electric field has to be applied at the point of nucleation, because the smaller structure itself, that alignment has to happen. It has been done by a group in Strasbourg, Kisopone, and they have made structures which is bridging the gap by electric field. The same group can be done, also shown that magnetic field can also align because the dipoles are different in this direction. It's possible.
Okay. Yes, exciting talk, Subhi, as always. I have two questions. One is, in one of your molecule, you have shown that you have ester group and amine group.
What group? First one?
One of your monomer, you have a ester at the top and there are diamine. So my question is it not possible that there is some sort of a covalent bonding while you are self-assembling them?
Not really. I'm confident about that. It's not there.
But have you done some NMR or something just to?
Yeah, obviously. We can always make it completely reversible. We can. Spectroscopic properties is also highly sensitive to all these changes. We are confident about that covalent. I can convince you easily on that.
Okay. Yeah. See, another question is, so when you grown this chiral material, enantiomeric material, then the growth was horizontal, surface to surface, right? Your secondary growth or secondary nucleation. Nucleation.
Yeah. In this case, it's completely orthogonal.
Yeah. So that again indicates that somehow either the chirality of the surface or at least the surface morphology playing some role in these.
So in the second case, we have ruled out this. We also thought about the surface chirality, which is helical pitch. It can have some kind of effects on this growth. Okay. So what we have done is we have taken monomers, chiral monomers, which is easily with a very less helix reverse penalty. That means left-handed can go to right-handed very easily. So what we have done is taken those monomers and tried to nucleate on the surface of this. Okay, of this. And if there is any chiral information from the surface coming, the resulting polymers will be chiral in nature. So we have exclusively got only achiral. That's one of the control experiments we have done, is to really rule out the surface chirality plays any role. So we have looked into it. And coming to this, I will explain in detail later. So we have ruled out the possibilities of chiral induction from the surface. So that we have ruled out, because that's one of the first questions, because we'd be really happy to see that. Because if surface catalyzed nucleation can be used to induce chirality, it's a zero chiral information you are getting. It's a symmetry break. You understand? It's a very beautiful experiment. That's the first thing we wanted to work. That worked that way, but that failed completely. So that is not there. We are sure about that chirality transfer from the surface. Second thing is the second case, chirality effects are very minimal. We have not used chirality at all as a controlling point there. It's all same chirality. There, the thing is that two different organization of the monomers, one is J aggregate and one H aggregate, and the surface is highly negatively charged. If you look into the database, so surface mapping we have done with Isar Mohali Gautam. So we try to, because this as there was a question, we were really worried about that 1 micrometer width. We wanted to know where is this charges coming from. So we see that surface mapping, we have seen that the charges are on this. So that is triggering the orthogonal growth actually. So there is no chirality in the second case. So I hope that convinces you that chirality is more.
Okay. There's a question at the back first, Mitro.
Yeah, hi. So I was just wondering, this might be slightly out of the scope of the talk, but I was wondering what specific application does the bottle brush kind of shape or morphology of the polymer have?
Yeah. So as I mentioned, so this preliminary, I didn't want to share it. So the kind of structures we work on are conjugated systems. So what we found this in this bottle brush is when we look into it, so when we excite here, okay, so there is an energy transfer to this one, and then you see an exciton migration. So this one Arindam has observed. The question is, can you attach anything here, a catalytic center or something, and harvest? I'm like, that's one of the things. But right now, I'm not worried about applications. We are just excited with the fact that we are able to control this topology in a precise manner. But obviously, this answer, I'm like, in few years, we'll address that applications.
Yeah. Thank you.
Yeah, fantastic stuff. I was intrigued by your goal of creating 2D networks and maybe 3D networks. And I was thinking how to make that possible, because what you showed was, okay, something is going out as secondary nucleation. But what occurred to me is that, is it not possible that the kind of secondary growths you have, they could have end-to-end coupling, in which case you have H-like formation. So one, secondary, H-like formation, and multiple such H-like formations with many things will give you a sort of a network.
Yeah.
Is that something that you see already? Yeah.
We have not seen it, because I think the concentration is the key there.
Yeah.
We have not seen this. I'm like, definitely we should look into it.
Instead of driving it through monomer addition, it will be then sort of gentle taking or something so that the fibers themselves add up. And might create a sheet or even a 3D structure, depending on how those things are.
So it's a nice concept. That I think the challenge we are going to face is the concentration, but then we will look into that. And the other things already I did not share, that what we are having right now is the layered structure also we are having right now. So it's again an ATP system, which forms a 2D sheet, and this surface acts as a kind of secondary nucleation. So this grows in both sides, and then sequential addition determines the layers thickness. So secondary nucleation is beautiful at this point. Like we are getting higher order structures with precision. So I didn't show that results. So we are able to now stack things in a layered manner. We have not done with the two different to make a heterostructure, but we have a same system. We have a layered structures, and that numbers determines number of sequential addition. So we have systems like that.
Right.
Yeah.
Just a last question. I don't want people to, otherwise we need refreshments right now, because it's late. Just this universal question that you might be getting everywhere, but what are your thoughts on this? Have you invested some time in looking at structure directly of these things, which can be done by different kinds of techniques that are available? The biophysicists, like just talked about cryo-TEM and other kinds of things. But have you looked at a thought of how to, at the end, there has to be a design principle that is making those 1 micron thick things with bottle brush structures, right? Yes, hypothesis is given, absolutely, and it works very well. You're controlling them very well because you have the hypothesis, and it's going according to that. But at the end, because there was a lot of work with proteins, hierarchical structures in proteins, people had to do the structural study, right? I agree. To get a little bit of, is there a collaboration, or is there a thought process of how you're...
we have not started. I mean, like, this is a right fact. I believe we have to definitely look into the structure. Even if I don't go to secondary nucleation, even a primary, whenever supramolecular polymers, it's very important to have that kind of structural analysis.
Right.
So there are people who invest time on it, on a different systems. Like a lot of SAC studies are done. I'm like, especially a guy from UK. So there are many people doing it, but then right now I have not collaborated on this. This obviously.
But do you think it's an important?
It's important.
Place? Yeah,
definitely is an important thing. Yeah.
Okay. Thank you, Subhi.
Yeah.
Let's actually end this talk. The session has not ended. We will have a break. We will take a break and we'll have some food, especially for Anukul, and coffee, and then we will move on to the poster session. Okay, and I request all my colleagues, faculty colleagues, and students and staff and everybody to come to the poster session. This is the heart of the department, the scientific work that goes on. You guys have to give time. Okay, come in to the poster session, listen to all the posters, and at 6 o'clock, the first poster session starts in. Actually, it is in the NMR building, the NMR ground floor, and the NMR first floor. Where is Malay? Malay's lab is coming up. So that room and the NMR ground floor. So please do join us.